we are moving right along. Our next presenter is Dr. Norman LaFrance. He is the Chief Medical Officer and Senior Vice President at Plus Therapeutics, and he was previously Chief Medical Officer and Senior Vice President at Jubilant Pharma Limited, and responsible for all pharma, medical, and regulatory affairs and activities. And prior to working in industry, Dr. LaFrance practiced medicine and held academic faculty appointments at Johns Hopkins University School of Medicine in the departments of medicine and radiology. We are very excited to have Dr. LaFrance speak to us today about a novel targeted radiotherapeutic for brain cancer, specifically the ReSPECT trials. Dr. LaFrance, take it away.
Thank you, Diane, very much. And I want to echo the compliments to everyone else. And first of all, your program, I'm grateful to be included in this with some other very talented folks, but more importantly, the comments we heard at the beginning on the journey people are on. To say inspiring, I think, is an understatement. But if we can advance the slides now, and I know one of your colleagues is helping me since I'm on a device that I can't share a screen. We have the obligatory forward statements, as others have said, other speakers have mentioned. I'll be talking about an investigational product. Next slide, please. And Plus, as an organization, I just want to spend a quick second. Our platform technologies are ideal for CNS indications, and particularly CNS primary cancers. There's direct tumor targeting, which I will touch on. The availability of the items to make the product are readily available with a secure supply chain. We all know CNS cancers are a difficult area and require this type of investigation. Next slide, please. I want to compliment and call out our principal investigator, Dr. Andrew Brenner, who started this program nearly a decade ago, and emphasized how clinical trials are important. And I've heard this, through the other presentations and people's comments, and perhaps it's the obvious, but it's my way to say thank you to folks who recognize and are willing to participate, not only for the potential benefit they may have, but for their contribution to new therapies, which in CNS cancers are particularly important and required. Next slide, please. We're talking about the CNS, and this montage of CNS activity, I think, underscores that people, I think, have heard that the last medical frontier is the brain. And maybe that's a little bit of an overstatement. There are many last medical frontiers, but it emphasizes how we have to be careful that the brain is a complex organ. It's involved in everything. You have a problem there. Besides needing to deal with that problem, you have to recognize and be careful about the rest of the brain's responsibilities. Next slide, please. And we look at clinical trials and why are new treatments important? Well, particularly in brain cancers, and thank you, Dawn, for mentioning my Hopkins heritage. I'm very proud of that, and had the good fortune to work with some very smart people. And folks like Skip Grossman, a friend of mine, recently got the Lifetime Career Award from the Society of Neuro-Oncology. Other colleagues that I worked with in the CNS cancers, particularly glioblastoma, Ben Carson, Henry Brem, Don Long, and others were real inspirations. And talking with them, and particularly Professor Grossman recently, we were reminiscing about the work we did, and we did some good work, and they continue to do phenomenal work. But our progress in defeating glioblastoma has been frustrating. We know a lot more about the tumor's biology, of course. Doesn't mean things haven't happened. But if we don't get into new treatments in the clinical trials, we're not going to be able to turn some of these discoveries in biology or some of the things we already know and improve upon those to make a difference. Next slide, please. So Plus as an organization really want to try to transform brain cancer from this poor prognosis into a manageable disease. Next slide. Everyone, I think, on the call is familiar with, how difficult this tumor is to treat. And over the decades, as treatments have evolved from surgery to adding some radiotherapy and some of the chemotherapy, there have been some incremental improvements. And in particular, radiation has been shown to make a difference. Radiation works. We know that in a lot of tumors. In my background at Hopkins, among other things, I had a lot of thyroid cancer treatments, and radiation there is known to have an important difference up to and including a cure. But you have to get the radiation to where you need it. And radiation now in glioblastoma, there are some very effective ways to deliver it, but that slide previously of the various brain structures, you have limitations. You certainly want to get all the radiation you can to the brain as possible, but recognizing you have to be careful and avoid off-target or normal tissue, normal brain toxicity. Next slide, please. So that brings us to our product and our trial. And the focus of this is the rhenium-186 isotope. It has a beta decay. I'm not going to get into the technical aspects of it on this presentation, of course, but we bichelate that for some stability and allows us to encapsulate that into a nanoliposome, which contributes to its delivery and to its retention at the tumor site. Next slide, please. And as you do with all new products, we had an extensive preclinical experience, a lot of information here. You'll have this as a resource document available, as Diane had mentioned at the beginning of our presentation today. But very briefly, you see that in the upper left, tumor retention, tumor response, and the survival in those three graphs are very impressive in the preclinical model and showing some imaging results of that, confirming very good preclinical, in fact, extremely promising clinical results. Based on this, next slide, consortium of folks, and I showed you Professor Brenner's picture earlier, he and his colleagues and 3 sites in Texas received NIH funding for a phase 1/2 trial, which is ongoing now. Next slide, please. And what you'll see with this is in the next few slides, what's occurred from this time forward. So the phase 1/2 trial has what you'd expect in a typical inclusion-exclusion criteria. These will be available in greater detail. It's over 18-year-old patients, although we'll be opening a pediatric trial by this summer, hopefully. And there's the other criteria there. Next slide, please. Importantly, and I'll spend just a few minutes, this trial requires the direct infusion of the encapsulated rhenium into the nanoliposome by a convection-enhanced delivery catheter, which is similar to, and these are inserted similar to a standard of care biopsy. But these convection-enhanced delivery catheters go right to the tumor, so you have a direct infusion of the drug into the tumor. And this slide outlines what's required for the treatment. So in the weeks before treatment, you have your reviewed screening, make sure that the treatment is right for you. Your physicians will review that with recent MRs, and for the catheter placement, which is already available with planning technology and MR planning, quite comparable to what many of you might have experienced in terms of the standard of care biopsy. We place 1 to 4 catheters. In fact, most recently in our dose escalation, and we'll have a slide on this coming up quickly. We've typically have now 3 or 4 catheters, and then the drug infusion takes place. It can be up to 4 hours, but often it may be 90 minutes to 2.5 hours, depending on the size of the tumor and the number of catheters we're able to place, which are all a function of the treatment planning, ensuring that we are avoiding any sensitive structures in the brain. And again, this is a well-established procedure, and any neurosurgical department certainly can take care of this. Either on the evening of that infusion, which begins maybe at 9 o'clock in the morning, for example, finishes by noon or shortly afterwards. The catheters may be removed that evening, and certainly by next morning, and you're discharged. Next slide, please. This is a very busy slide to point out. The study's been ongoing. We have nearly 30 patients have participated in the dose escalation. We have in one of the dose, a non-DLT dose, we've started a phase 2 for small to medium-sized tumors. That's ongoing and has nearly a half a dozen patients entered into that trial. I won't go into these details here, but the trial is well underway and is proceeding very nicely. Next slide. Importantly, the product is very well tolerated during the procedure. The catheter placement probably is the most uncomfortable. It's a surgical procedure, so the insertions per se are well tolerated. There might be some scalp discomfort. If folks have had brain biopsies, you can probably appreciate that. And the adverse events are typically non-serious, and the few serious ones that are here were not product related, and some of these were actually pre-existing conditions. For example, lymphopenia was pre-existing before our treatment, and the cerebral edema might have been due and likely was due to too rapid a steroid treatment, steroid taper after treatment. Next slide, please. I'm going to finish up the talk by going over some examples and some of the data. Here are 3 examples. I mentioned the direct delivery. You see the MRs, and then that second image on all 3 rows there represent the SPECT image, which besides the beta particle, which is the therapeutic particle for the tumoricidal activity, there's a simultaneous gamma ray, which gives us real-time validation of where the product goes. And although I'm just giving you one image, this delivery is complete within a few hours. That 24 hours has not changed, and we have, with the investigation images, through 7 days, which shows this exact same distribution. And you can see some of the benefit that's occurred. Next slide, please. And here I spend just a quick time showing the very provocative preliminary evidence of some promising efficacy. And this is reviewing the types of things we're doing in the dose escalation trial on the left, but more importantly, the overall survival curve. And this is very brief, and it may be difficult to read, but then you can go into either our website or go on to Delana will have this available, as she said earlier. This is the Kaplan-Meier curve within our trial of patients who received less than 100 gray, which very early in the dose escalation, there were patients that had that lower delivery because, the phase 1s are primarily related to safety first. But radiation we know works, and we're finding even in the phase 1 dose escalation, as the dose is going up, we're getting higher dose deliveries to the tumor. And you can see in those patients who got greater than 100 gray, and this follows exactly the same results we saw in preclinical studies, which is very encouraging if we're replicating in people what we've seen in the preclinical studies. And you can see, hopefully you can see in the bottom graph, that those greater than 100 gray had a 76-week median overall survival versus those less than 100 gray with a 22-week overall survival. And those numbers, this is within our trial, but the 22 weeks is not a lot different than, say, bevacizumab or other convection-enhanced delivery overall survivals in control groups, which has been typically around 30 to 32 weeks. Next slide, please. So in wrapping up, next slide, please. Another example, finally in our phase 2, you can see the brainstem, and the tumor in this location is in a very difficult, deep area, was not surgically accessible after the original surgery, obviously a recurrence. We treated this patient in the phase 2. You can see some of the MR slides, some MR images later on that have made a difference. We have good tumor coverage, 105 gray absorbed dose coverage, both of which, in the 3 months since this patient was treated, has remained stable and actually improved nicely. So next slide. So, I'd like to end here on showing another example in the same patient of that activity. And now you see the SPECT image with that retained retention. Next slide. And so I'll turn this back over to Delanne and answer any questions. But particularly if you feel this trial may be of interest to you, please contact us, talk to your physician. We're on clinicaltrials.gov. Delanne can give you contact information. You saw on the beginning slide my contact information. And I'll stop here, Delanne, and turn it back to you for any questions.
Awesome. Thank you, Dr. LaFrance. One of the things that I would like you to explain to our audience, because sometimes we all use some slang, right? We all use words because we're using and talking about this all the time, right? So if you could go back and talk about CNS and what that means. I know what it means, but what does that mean to our general audience?
CNS, well, central nervous system, and in particular, they're we're interested in the rare tumors of the CNS, and particularly ones that don't have really effective treatments. And glioblastoma is, I think, right at the top of that list of challenges and unmet medical needs. It's been studied a lot. I told you about my experiences with colleagues at Hopkins in the past. We did a lot of good work. They've done tremendous work since I left decades ago. And but quoting them indirectly, it's been our frustration that we've not made the progress, particularly in glioblastoma, that we all wish we had made. We know a lot more about that disease than we did 20 and 30 years ago, but have not been able to translate that into successes, because the successes we've had, for example, the chemotherapy, well, you have diffusion issues and it doesn't stay where you put it. The convection enhanced delivery catheters are developed for that direct delivery of chemotherapy, but those molecules haven't cooperated in staying put. We've taken some of those advances, the success of radiation, but having to be careful with how radiation now is delivered, and it has to pass through normal brain. They do a good job. It makes a difference, but there are limitations of how you can use it. What we've done is encapsulate that radiation into a deliverable a nanoliposome, directly deliver it to the tumor, have shown on the slide, hopefully, and we don't have the time or detail to do that, it's durable retention in that area, so that radiation stays put, doesn't affect the normal tissues or the normal brain, and delivers its payload to the tumor and where it's needed. And for example, typical delivery, radiation deliveries, external beam radiotherapy as an example, 30, 40, 50 gray, it has to be fractionated. It does make a difference, but once they get to those levels, they're kind of finished, because they, it's not first do no harm, but in many ways, that's exactly the philosophy. You don't want to cause more problems. You got to do all you can, but you also have to respect. Think of the slide I showed you with those, the brain activity is very complex. Areas of the brain are very radiosensitive, normal brain. And we know that radiation is tumoricidal and it works. And if you can get it where you need it, it works. And our preliminary data is showing this has real promise. And that's with a single administration. I didn't spend any time now, but our expectation is where our safety profile is so robust on this product, we're anticipating going to a multi-treatment paradigm with patients. That's to be determined, is to be reviewed with FDA. But because of what we're getting now, we're seeing that that's a real possibility. Hence the slide I showed that, it wouldn't be fair to say, oh yeah, this has potential to cure neuroblastoma. That's a little bit too bold. But if we can give this on a repetitive basis, and our data shows that we can do that, some retreatments, and it's very well tolerated, this may be one way to accomplish that.
Right. It might be a tool in the toolbox.
Exactly.
Yeah. So I know that Plus Therapeutics would like to be able to bring this clinical trial to other sites across the United States. So if there are healthcare providers listening in, and you are interested in thinking about opening this clinical trial in your site, I encourage you please to get a hold of Dr. LaFrance, because again, it is all about having multiple tools in your toolbox to treat disease. So thank you. Are you at all able to, and I know some things you can't say, but the pediatric clinical trial that you're hoping to open up, are you able to share a little more information about that?
Well, high-grade gliomas, ependymomas in kids, it'll be under 18. We'll probably start about age 6 because in an abundance of caution, there's a developing brain, and usually is about as developed as it will be in terms of increased radiosensitivity by 4 or 5. So our first focus will be age 6 up to 18. And there'll be actually some overlap above 18, depending, a lot of kids have survived, say from a teenage presentation to older. So there'll be a fair amount of flexibility. There'll be high-grade glioma and ependymomas.
Wow, that's wonderful.
And we'll be talking to you and maybe getting it on your site once that gets started. Of course, it'll be on our website, and it'll be on clinicaltrials.gov. So, yeah, thank you.
Yeah, you're welcome. Thank you.
If you allow me, I'm sorry. And this same product, although this is not the, for those who are involved in managing leptomeningeal metastases, this product is also in clinical trials for that via a direct administration via an intraventricular catheter, Ommaya reservoir, and that's an outpatient procedure.
Okay.
So thank you for that. Allow me to say that.
That's very important. Thank you.
Thank you.
Okay, we have just a couple minutes. If we have any more questions for Dr. LaFrance, please ask him now.
This is Jen Ortiz, and I have a question.
Yes.
Dr. LaFrance, thank you for being here today. I'm a 2-time glio survivor, and I was curious if your current research includes DNA from the tumor as a part of that research for trials. And I ask this because I'm curious if this will play a part or a role in the outcomes for the masses when comparing the data in the future.
Yeah, that's a great question. And our current trial is not looking at that, but I'd love to hear some more from you on that. Our and it depends on if you're looking at the tumor DNA but our leptomeningeal trial is actually looking at DNA damage from the radiation as part of a separate potential investigation. I can't go into the details on this, but my email was there, and if you'd like some more information, please communicate with me, or Deland will certainly connect us, and I can go over into that detail.
Thank you, doctor. I appreciate
You're welcome.
Time.
Yeah, great question. Thank you.
We have time for one more question if it is out there. Okay, wow. Well, thank you so much for your presentation, Dr.
I appreciate the opportunity. And again, I appreciate everyone on the call, and you're all an inspiration for me. And this is a terrific presentation and day. Thank you.
Thank you. Well, it's a team effort.
Yes, it is. Yes, it is. Yes, it is. Well said.
Thank you.